Good evening, ladies and gentlemen. I'm Bill Roper, and it's my distinct pleasure to welcome you to the 2010 Norma Berryhill Lecture. This is a major event each year in the life of the UNC School of Medicine. We celebrate the opening of our fall term. This is, in effect, the fall convocation of the school. It is our start of the year. It reaffirms our commitment to scholarship, to our students, to our faculty, and to the very collegiality of this place that the Berryhills so represent to this institution. This lectureship recognizes a highly cherished individual, Norma Berryhill, who, along with her distinguished husband, Dr. Walter Reese Berryhill, contributed immeasurably to the development of this institution, to the recruitment of an outstanding faculty, and to the enhancement of the collegial ambience that characterizes Chapel Hill. Indeed, Norma and Reese Berryhill hold an exalted place in our School of Medicine's history. Reese Berryhill, for his monumental accomplishments as medical school dean from 1940 to 1964, which included the expansion of the medical school and the building of the North Carolina Memorial Hospital, and Norma, his partner, for her love, loyalty, wisdom, strength, and support. I know they would have been pleased to know that we recently announced the expansion of our medical school to regional campuses in Charlotte and in Asheville. Mrs. Berryhill was a principal architect and engineer, as someone has called her, for the School of Medicine and for the Medical Center. And she nourished generations of students and provided wisdom for all the deans and for the faculty, and for this we are tremendously grateful. I'd ask you please to join me in offering our gratitude for all the Berryhills have done for us. I'm pleased to recognize members of the Berryhill family. Kat Williams, their daughter. Please stand, Kat, so we can recognize you. And Jane and David Mayer, the granddaughter, and her husband David, please. And I'd like to recognize my distinguished predecessors, Dr. Stuart Bondurant, who was the 1993 Berryhill lecturer. Stuart. And Dr. Jeffrey Haupt, who was the 2009 Berryhill lecturer. And finally, we have a number of other previous Berryhill lecturers. Would all of you please stand so that we might recognize you? Previous Berryhill lecturers. It's my pleasure now to welcome Chancellor Holden Thorpe. He's here to bring greetings on behalf of the university. And Holden, it's a continued delight to have you so deeply involved in the life of the medical school. Please come join us.
Thank you, Bill, and thank you for inviting me to be here today. I'm really excited to see all of you and to hear Jenny's talk, and so pleased for you, Jenny, that you were selected to give this lecture. It's entirely appropriate and exciting honor for you, and you represent so much of what this medical school and our faculty are committed to in terms of outstanding scholarship, service to the institution, and to our students, and so it's a privilege for me to be here. In fact, I'm so excited to be here that I've actually come to this talk twice. We process a lot of invitations and things in the Chancellor's office, and we're human. So I was here precisely at this time last Wednesday, and none of you were here. And because this happens from time to time in my life, I carry around a set of note cards that I send to people when I miss something because of a scheduling conflict. And on the front of the note card, there's a picture of a man in a suit, and he's holding a briefcase, and he's talking on his cell phone, and he's standing in the redwood forest. and the caption is, "Carol, I think we need to check the appointment book." So fortunately, because I didn't miss this event because of this. I didn't have to send Bill one of those cards, but that's why I keep them around. It's been a great year for the School of Medicine. I think the plans for the regional campuses in Charlotte and Asheville have matured, and we're very excited about implementing those. They bring great recognition to the university among our colleagues in the General Assembly and in the UNC system, and so we appreciate your willingness to participate in that. And of course, it was an outstanding year for research funding, $803 million for the university, and the School of Medicine scientists account for more than half of that. We're pleased to see the brick building going up. It's possible that when I cut the ribbon on the brick building, there might be a lull before I cut the next one. That's going to be the last project that we will get through for some period while we wait for the economy to straighten itself out. And Bill and I and so many of you worked so hard to get that done. But it'll be exciting when we dedicate that building, and just so rewarding for me to be part of what you do in medical education and in science and caring for our patients. We had a wonderful occasion dedicating the piano for the cancer hospital. Bill and Shelley and I were there, and Terry Houston, who's our interim chief diversity officer, sang a few great American standards, and I played your piano, which is wonderful. And then Tommy Otten from the music department, who's a real piano player, played a Chopin nocturne. And to see the cancer patients and the administrators and the physicians and the chief diversity officer and the chancellor and the music faculty all coming together and mashing up all of these wonderful things that we do at the university in that way was really, that's the kind of thing that makes my job so much fun. So thank you for inviting me to be here tonight, and congratulations, Jenny.
Thank you, Chancellor. It's also my pleasure to introduce Dr. Joe Jenkins, who's the president-elect of the Medical Alumni Association. Joe, come bring greetings.
Thank you, Dr. Roper. It's a pleasure to follow Chancellor Thorpe, particularly in these days when most people are either hiding behind him or trying to avoid him. And I want to commend him for navigating the university through a difficult moment, very brief moment in time. When I travel on an airplane for more than 2 hours, I usually take my throwaway journals, and most of you know exactly what they are. They're shiny journals, and they come in a variety of the names depending on what your specialty, what your training is. Mine are Urology Times or the AUA News, Oncology Times, Renal and Urology News. And on a flight recently, I was doing my catch-up reading with these throwaway journals. And the first one I opened, I believe, was Oncology Times. And I read the headlines, and I went, wow. And the headlines was this: Duke Scandal Rocks Research World. Now, I've got to admit that for just a tiny instant, and Dr. Colin Thomas will remember this, I was a little bit like the neurosurgeon in our surgical textbook during our residency, who said that the heart of a neurosurgeon leaps up at the sight of a meningioma. Well, for just this brief instant, as a Tar Heel, my heart leapt up at the thought of problems at Methodist Flats across the way. But reality very quickly stepped back in, and I realized it could be any of us, could be any university. If you don't know what happened at Duke, an associate professor of medicine and a genetics researcher fabricated an element of his curriculum vitae. He claimed to be a Rhodes Scholar. He was not. Worse, he published in very respected journals, including the New England Medical Journal, research that focused on how to genetically identify which lung cancer patients would respond to which chemotherapy agents. And that was a hot issue. The researchers at Baylor and the Mayo Clinic, however, or the MD Anderson and the Mayo Clinic, could not reduplicate his results, verify them, from the methodology that was published. And so it brings us to the timeliness of the lecture tonight, the Berryhill lecture that Dr. Ting is going to present, because it reminds us how intensely focused and important research is. Now, why is that? As medical students, we thought, oh, God, that was terrible. But we all did it. And we listened, I hope. You just heard Chancellor Thorpe talk about the research monies coming into the university, over which half of that $823 million is coming into the medical complex. So we're talking a very important amount of money here. Secondly, you better than most people that are gathered understand the importance and the prestige of publishing a breakthrough finding that will lead to better health care. And third is, in this next decade, the key word and the key phrase is translation. How do we translate your bench work, your lab science, into faster clinical care, so that the patients in North Carolina, the patients in the USA, and indeed the patients in the world benefit? And so it is very timely that our lecture tonight is about the subject that Dr. Ting has chosen. The word that we take away from it is integrity counts. The integrity of the process, the integrity of the methodology is critical to our success as a university and as a people. And it's not just an undergraduate school, it's not just football players, it's most importantly in our labs and in our medical school environment. And so, on behalf of the Medical Alumni Association and its graduates, who I hope understand the importance of methodology, it is delight. I welcome all of you here, and we are looking forward to our speaker.
Thank you, Joe. It's now my pleasure to introduce our speaker, the 2010 Norma Berryhill Distinguished Lecturer, Dr. Jenny Ting. I hope you've taken the occasion to read her very distinguished biography in the brochure for tonight's lecture, but I'll just summarize a few points if I may. Dr. Ting came to UNC after doing graduate work at the University of Southern California and Duke in microbiology and immunology. While at Duke, she decided to study why cells express certain genes but not others. After 2 years there, she was recruited to UNC, where she's been on the faculty for the last 20 years, and she's made a significant impact on understanding how genes are transcribed in the human genome. Apart from the very important research work that she is conducting, she also has a passion for teaching and for mentorship. Her chairman, Dr. Bill Goldman, chair of microbiology and immunology, says that in addition to enjoying getting her students excited, she, quotes, engages colleagues in productive collaborations and influences many projects outside of her own research interests. And I've heard that from a number of you, even tonight, as you've talked about your joy at her being this lecturer this year because of what collaborations you've had with her. Marcia Runge, now Executive Dean of the Medical School, has commented that Jenny's leadership has made it possible for us to recruit outstanding scientists across the board and she makes every effort to help others be successful. She's been recognized by various scientific organizations, including serving on the advisory board of the National Institute of Allergy and Infectious Diseases at the NIH. She's received the NIH's Individual National Research Service Award. And most recently, she was honored by the National Multiple Sclerosis Society Volunteer Hall of Fame for scientific researchers. Ladies and gentlemen, please join me in welcoming Dr. Jenny Ting, our 2010 lecturer.
Thank you so much for such a generous introduction. Dean Roper, Chancellor Thorpe, the Berryhill family, honorable guests, my dear colleagues and friends. So it is indeed a greatest honor to be here to give this opportunity to give you the lecture today. I'm especially honored to receive this distinguished honor because I'm humbled to look at the list of previous recipients. This includes directors, deans, department chairs, and a Nobel Prize winner. I don't know if I can match any of these. In addition to that, you will also notice that I look a little different than many of the previous recipients. I'm at least a foot shorter. So to get back to this, when I was selected for this lectureship, I must have received 10 different emails from all my female colleagues to congratulate me and to tell me that I represent many of them. And so this is indeed a great honor. I should say, prior to my selection, however, there has been another female lecturer for this lecture series, and her name is Mary Ellen Jones. For those of you too young to remember her, she was the chair of biochemistry when when it was very rare for chairs to be female. And she was a National Academy member, and she was a mentor to many of us young faculty. In addition to that, she has a building named after her, and I'm pretty sure after this, if I request that, it will be a very difficult thing to match. So I don't see too many buildings around here without a name yet. I think Mary Ellen, in her very understated fashion, but effective way, has really nurtured her department and nurtured many young faculty, including many of us. And she best exemplifies what is good about Norma Berryhill. So I took my opportunity to really learn about Norma Berryhill, and I just couldn't believe all the things that she has done. As Dean Roper has mentioned, she was the wife of our first and founding dean, Dr. Berryhill. She was certainly considered as a co-founder of our medical school. And one of the things that she has done is she realized that when faculty or students or the staff come, they come with their families, and frequently the families have left their own comfort zone. And as a result, they really don't have a social support system. So what she has done is visited these families, and I have a feeling some of you might be from that era, and I just basically couldn't believe all the things that she did. She made dinners for people, and supposedly she has, from what I read, she has baked hundreds of casseroles to deliver to the faculty and staff. And I think, as somebody who cooks, just for that, she deserves the Medal of Honor. But what we tend to forget about Mrs. Berryhill is that she was accomplished even before she was married. That she actually graduated from Carolina, is a real Carolina girl, that she actually was the director of a girls' club before she went on to graduate school and went up north to Columbia and had graduate training. And subsequent to that, she came back and became the dean of the girls at the Central High School. So all this was accomplished before she married Dr. Berryhill. And after that, when she came here, and this is, of course, the picture of her. And when she came here, she organized all these associations and clubs for the wives of the medical students and as well as the staff. And at that time, from what I learned, it was difficult to actually recruit people to come to Chapel Hill. And I always wondered when after I came to UNC Chapel Hill about the uniqueness of our school, that we have such a collegial, friendly environment among all our faculty. And I always wonder who started all this. And in reading about this, we learned who really was the originator of these very collegial attitudes towards one another, and that is Norma Berryhill. In addition to all the things that she did for the medical students and staff, she also organized the Auxiliary Association, which is now continued as the volunteer service and association at UNC Memorial Hospital. So I think her legacy has continued for over 50-some years. And in the school was started in 1952, so I think in another few years, actually it's already the 50th year anniversary and past. So her legacy has continued for all of this time. Now, I will start this lectureship by telling you about a story. As many of you know, many of us give a lot of lectures and seminars at different places. And one of the rituals of the seminar giving is we have what you call the lunch with the graduate students. And this is very important because the graduate students get a free meal. So they all come and talk with us. In addition to that, they also get a perspective that's from the outsider. So I went to one of these luncheons, and I was sitting down with this graduate student. And this group of students were asking me about my own career. So I said, well, I actually went to UNC over 20 years ago and have been there since. And he looked at me stunned. And this graduate student, who doesn't seem like he has a filter, blurted out, "You mean you have nowhere else to go?" And I was very astonished because I thought this is just normal. And so I proceeded to tell him that actually most of the colleagues who were recruited at the same time that I was are still at UNC. In fact, on our floor, we have people who have been there for 5 months or people who have been there for 30 years. And we have all maintained our allegiance to UNC. So I told him all this to show him that we are neither weird nor undesirable. And I emphasized to him that many of us probably could have gotten a job elsewhere if we really wanted, but we don't. So we are here at UNC for a very long time. So I think this is one of the unique features of UNC, and when I recruit new faculty, I always tell them that we have a lot of world-class scientists, but we don't have world-class attitudes. And I think that's something very unique about our school. Now, my own start at UNC came when I was fortunate enough to be interviewed for an immunology position at UNC. I did a postdoctoral training with Dr. Jeff Frelinger, who was the chair of microbiology when I was at the University of Southern California. Subsequently, I left his lab and came to the Triangle due to a position that was offered to my husband. My second postdoctoral position was done at Duke, and I'm using all my diplomatic training to say that it was a very challenging time. But to this day, I really have to thank Providence for the turn of events, because what happened was a year later after I arrived, Jeff was recruited to UNC, and he learned about my circumstance and really encouraged me to apply for a job that opened here in the Lineberger Cancer Center and at the Department of Microbiology and Immunology. So I was fortunate to be selected as the final, probably, applicant, because I came, gave my seminar, and I'm sure Joe remembers, the next morning he called me and offered me the job. I think I was so stunned that I said yes, and I didn't negotiate for anything. And I didn't know what the job was about. I just knew it was a tenure track position, and I was so happy. I was jumping up and down, and soon thereafter, news traveled down the hallway, and that afternoon, the chief of our division came and said, we actually also have a position now for you at Duke that's also on the tenure track. And I politely told him that I gave my word to Joe, and I can't take my word back, so I have to come to UNC. But to be honest, deep down inside at that time, I knew this was a much better fit for me. And so to this day, I'm absolutely delighted that that was the decision that I made. So one of the true joys of coming to work here is the interaction with all my colleagues. And so I have actually gone back to my CV and listed the people that I've collaborated with the publication or with shared grants and with recent papers or projects that are ongoing. And I really apologize if I left anybody else out, because by the time I got to the end, I can't remember where I started. And there's over 60 labs that we have collaborated with. So in many of these cases, what happened is we went to the lab and seeked advice in a field that we know very little about. And on the other side, we were received with generosity, and people have really taught us many new fields. And so this is a list of my collaborators, and I'm sure there are many that I have actually missed. And as Chancellor Thorpe has said, our federal grants at UNC has actually gone up substantially. I went back to one of your speeches about how much it went up, and it went up 13% from 2009 to 2010. I think the reason for this is clearly because we're interactive, we're really ready for collaborative research, and as a result, we're super competitive in this very competitive era. In addition to that, as a young faculty, and now as a not-so-young faculty, we've also benefited from the steady hand at the leadership. So I have benefited, and many of us, from the incredible leadership of Joe Pagano, followed by Shelley Earp at the Cancer Center. And I have received really incredible support from our department, headed initially by Fred Sparling, and then by Jeff Frelinger, and now by Bill Goldman. And I've also, as well as many of our colleagues, benefited from our association, in my case, with the genetics curriculum, as well as the neurobiology curriculum. In the last 2 years, however, our former Vice Dean, Dr. Etta Pisano, has convinced me through a very arduous process that I should start to take up a leadership position. So with my colleagues, including Dave Peden here, we've started the Inflammatory Disease Institute, and I hope we can emulate many of the leaders that I have just mentioned to promote inflammation and immunology on this campus. So of course, for those of us, the title of my talk is about joy first. Our greatest source of pride and joy is our work, our body of work that really defines us in our professional career. So I would like to take a few minutes to describe our work at Carolina. This work, however, could not be accomplished without the list of trainees who have gone through my lab. And again, I was a little stunned because I didn't realize I have been here for this long and trained this many people, and I instantaneously felt a lot older. So among this group, there are so many accomplished previous members of the lab. Out of this group, there's a director of a big lab at NIH. There are 2 chairs of departments. There are many tenure track faculty, and there are many other faculty at UNC who have contributed enormously to the success of our universities. Among this group of people in the lab, they have also been productive both within the lab as well as outside of the lab. So they contributed to nearly 200 scientific papers, and while they're in the lab, while during their tenure in the lab, they have produced over 40 babies. So this is the boys and the girls, about equal distribution on both sides. In fact, I divided that by my tenure at UNC. I think that's more than one a year. So on the right-hand side are the current members of my lab, and this includes junior faculty, postdoctoral fellows, students, our technical staff, the two people who really take care of us, and our administrative support, Mark Kramer, who's the assistant director of the Cancer Center, as well as my able assistant, Jennifer McFarland, who takes care of all of us. So in the following few slides, when I'm introducing my talk, when I say we, it really is this group of people, all these people, and not really me. I'm just a cheerleader for many of their successes. So when I came to UNC, I was very, very lucky, and I had, immediately when I came, I had a lab full of people and 2 grants. So times were a little bit easier then, so I don't want the new faculty to get depressed. But in those days, it was quite a bit easier to achieve a grant success. To this day, most of my later research really stemmed from this early work. However, we have approached this in a very attention deficit fashion that is very hard to trace what we have done. So that's what I will try to do in the following about 10 minutes. So one of my passion is really understanding this molecule called MHC, and this stands for Major Histocompatibility Complex Gene. And this protein, the protein that was encoded by this gene, was initially discovered because it causes transplant rejection. And this is one of the most polymorphic genes in the whole human genome. And each one of us have about 14 different genes encoding for this entire family. So the chance of your MHC protein matching mine is almost nil. And for this reason, we have the problem of transplant rejection. However, the existence of these proteins is not to make transplant surgeons rich, but there's another reason. And the reason is because these proteins are actually the recognition molecule for another molecule on this cell type called the T cells. And these are the T cell receptors. So this recognition is what activates the whole immune part that we call adaptive immunity. So this is what's important, for example, for vaccine, for when T cells still remember that one has been vaccinated 50 years ago. And now when you encounter the same influenza, for example, the memory is back. And this is the process that really determines this whole process. So I was really interested in this protein, the MHC. So like I said, when I came, I was fortunate to have received 2 grants, one of which was really about this protein and its functional role. There were many work done on the immunological function, but at the time, before I came to UNC, in Jeff Frellinger's lab, we found that this protein was also found in unexpected places, such as in the brain. So this result was actually met with a lot of skepticism, and as stubborn as I am, and I was then, and I still am, we decided we really want to pursue this to the end. So what we discovered was that this molecule MHC class 2 in the brain has a very, very major function, and this is work done by Glenn Masashima, who is now a faculty in the neuroscience department. And what we did is we were able to get mice that don't have MHC class 2 and those mice which did. And we compared them in a neurological disease that's genetic in its origin. So this is a disease that turns out not to be dependent on the T cell component. So this is not an immunological disease, but a disease of the CNS. And we found that this protein, which is normally thought of as only restricted to the immune system, actually has a very profound effect in the CNS. And this is shown here. When we compare these 2 mice, we found that the mice without this gene actually twitched a lot less because the white matter is in much better shape than these mice. And this is a mouse without the gene, and if you look at the mouse with the gene, it's twitching like crazy. So this is very blurry, and you can't see it, and this paper was published about 11 years ago. So this is the start of what we have done, and in 1995, actually it was published about 15 years ago, we said in our paper that this study really has very broad implications from what the immune system can do in other organs aside from the organs that we typically think of as being lymphoid. So we said this study is pertinent to an array of non-T-cell mediated neurodegenerative diseases in which class II MHC expression expressing immune cells are elevated in the CNS. It shows for the first time that these molecules have a profound effect on the course of neurodegeneration. And this has implications for other neurological diseases with similar features such as Alzheimer's, Parkinson's, and Huntington's. I think this statement is pretty prescient in that right now there's a lot of research actually into the inflammatory and immune etiology of these diseases that we typically don't think of as being immunologic. So in the Alzheimer's field, one of the very exciting studies are work trying to understand what are the roles of these cells that express this gene in promoting Alzheimer's. So I think this paper was quite significant in what it said. From there, we went on to look at other immunological molecules that might affect the CNS. And one of our studies showed that remarkably, the inflammatory molecules have both positive and negative roles. For example, in the case of this molecule called TNF, and this is the molecule for those of us who have arthritis. This is what is being targeted for drug therapy. So if you're taking Humira, or if you're taking etanercept or Enbrel, this is what's being targeted. However, none of these drugs can be used for patients with multiple sclerosis, which is a CNS disease. And we think the reasons is as follows, because actually this molecule can be good in the CNS. So this is what we found. We found in the wild-type mice, we have lots of reparative processes that are going on in the CNS of these mice. So this is progenitor oligodendrocytes. They're very proliferative in this disease model, and in the knockout mice, we don't see many of these progenitor cells that can repair the white matter tract. And this seems to be going through this receptor, the TNF receptor 2. So there's quite a bit of nuance in thinking about inflammatory connections to different diseases. And we found that this is actually a beneficial thing, and one should not stop this molecule. And so in the clinic, actually, this is correlative with discoveries that many of these molecules that are used to target this pathway actually exacerbated multiple sclerosis. So we provided the rationale and mechanism for this discovery. Subsequent to that, we actually found other molecules which are not so good, and so they actually destroy the white matter tract, and they're not important for repair. So some of these molecules are actually being tested with major companies to see if they can be used for translational research. Now, the second grant I received was trying to understand how genes are regulated. So before I went to Duke, I really did a lot of soul searching because I decided that what I will be doing for my postdoctoral fellowship should be what I wanted to do for the next decade or so. And when I was at Duke, I was given actually total freedom to do whatever I want as long as I can establish molecular biology within the department. So I decided, since I'm interested in this gene family, that I really wanted to know the promoter system. The promoter is the DNA that regulates the gene. And so when I came here, I was fortunate to be joined by a large group of postdoctoral fellows, and we published the first paper showing this promoter that is common among all 14 different MHC class II genes. And Trish Basta, who's in the audience, was the first one to really define this promoter. At the time, I was very out of the loop, shall we say, and quite naive. So I didn't know that there were 5 groups two at Harvard who were also trying to do the same thing. So we naively published this paper, and years later, one of the postdocs from that group, who is now a colleague of mine and a very good friend, told me at the time, his mentor, who's a professor at Harvard, said, "Who the heck is this Jenny Ting?" Except he didn't use the heck. He used some much more colorful language that didn't even start with H. But so we published this at the time, and then soon after, we and others in the field started to understand the DNA binding proteins, and we helped define some of these binding proteins. And one of the seminal molecule, however, was discovered by another group. And this is the master regulator that I will tell you quite a bit about in a few slides, the class 2 transactivator, or C2ta. But through the process, we and others, I should say, identified many of the proteins that can regulate the promoter. And another group identified this central master regulator that's a remarkable protein in that if you pop it onto any cells, it will express class 3 MHC genes. So this is truly remarkable and truly a very specific protein. This enucleating factor then also recruits many of the histone-modifying proteins, and these are proteins that really change the structure of your high order of the DNA. And so this is a nucleating factor that's responsible for all this, and all of this is covered over a 10-year span of work involving a multitude of grad students and postdocs. So from here, I decided about 10 years ago that these are really great studies, but we wanted to explore new territories and new directions. So we took on several exploratory projects to find out what other new genes we can find in the inflammatory pathway. This is also known as the fishing expedition. And if you do this kind of research or propose it in a grant, it's the kiss of death. So we never proposed any of these in grants when we were starting this research. And so I will tell you about 3 different directions that we have undertaken. So the first one is the question, since C2ta is such a good protein that causes the expression of all these wonderful genes, does it have other targets? And so we did a genomic approach to look at all the genes that might be regulated by this. And our outcome is that there was a gene called plexins, plexin A1, that was regulated by C2ta. The plexin caught my eye because I also work in the nervous system. And plexins are very important neurologic axon guidance molecules. They are important in telling axons where they need to go during development. So if you have a wrong move, you might not be able to sense properly. So these are clearly important in the CNS, but the question is, what is their role in the immune system? And the first one that we defined, we found that this has a very important role in linking with another molecule on this cell, the T cells. And this pair is important for the activation of the immune system. We also found that this is important for forming a proper cell shape through a signaling molecule called Rho, leading to changes in the actin proteins. So then we decided, since we found one plexin molecules, there are a multitude of plexin molecules that are known. So which other ones are known in the immune system? So with this, we did another search and found 3 different members of this family, all of which are found in the immune system. So I will just very briefly summarize 5 years of work done by 3 talented people. And what we have found is an embarrassment of riches when we were going through these exploratory projects. In the first one, we found that plexin A4, we were able to obtain a plexin A4 knockout mice from our collaborators. And what we found is that this knockout has a very significant drop in its ability to signal the cells. And this ability to signal the cells through this pathway called GTP-RAC leads to a loss of cytokine production in response to bacteria. And the loss is about 75%, which is exactly what we wanted. So this reduced this very inflammatory state that is probably not good for us, but retained enough immunity so we can defend ourselves against infections. And so we took this mouse and went forward and did a large number of tests And the test that's most relevant is we actually use a test called fecal ligation. And this is where you actually get bacteria to leak out of this intestine. So they're coming out of the intestine, and therefore you have infections in the peritoneal cavity. And most of these mice actually suffer through this process. But the knockout mice, most of the wild-type mice suffer through this process and die. But the knockout mice did much better. And this is very similar to what is called sepsis, where people come with enormous bacterial infection, and because of a huge inflammatory response, they die from that response. And here we show that when we removed this gene, we really ameliorated death. And we now have made an antibody that we're in the process of trying to see if we can actually ameliorate this huge inflammatory process called the cytokine storm, and if we can reduce death as a result of sepsis. I should say that as of this day, the only treatment for sepsis is try to give the patient antibiotics. After a certain point, there is really a point of no return, and we're hoping that we can do something about it. In the second plexin gene, we found this also regulates signaling pathways, and we find that remarkably that this protein actually changes the way cells move because it alters this. And the third plexin gene, plexin D1, we found that it is very important for cells to know when to move to a certain compartment. And this gene is expressed by lymphocytes called the B lymphocytes, and these are the lymphocytes that make antibodies. So they are the lymphocytes, again, that are important, for example, for vaccine efficacy. And what we found here, I don't know if you can see this, but thank you very much for turning off the light. What we show is this is the wild-type mice, and if you look at the B cells, it's all over the germinal center right here. And this knockout mouse, there's really a hole, so the B cells actually cannot move into this very important compartment that's important to develop B cells, and that is this germinal center. So the absence of this actually makes the cells defective in their ability to move into the proper compartment. And these mice actually make very little antibodies in response to vaccines. So our second exploratory project was a different direction. That is, we found this gene, the C2ta gene, which encodes a protein that has a nucleotide binding domain and the leucine-rich repeat domain. So we wanted to know if there are other genes in the genome that look like this. And I have to give credit to two people in the lab who thought about this. And they went and used this approach of two domain searches and scanned the human genome in the late 1990s. And this was before the human genome was published. And as a result, they were able to painstakingly find 22 proteins which all have the structure of MBD and LR. And what they were amazed by, they would come to my office every other day and said, I found another one. So after about 6 months, we completed the project, and to this day, this is the entire family. So their bioinformatics work was really absolutely beautiful. And what was amazing was that this family of genes are actually preserved in plants. In fact, later we discovered that Jeff Dangle in biology works in this family, except in plants. So this is evolutionarily conserved all the way from plants. The common weeds that you see on the street, they have these genes to us. So this is, to me, truly amazing. So we thought that these genes must have very important functions because they're so preserved in nature. And we decided very deliberately to change the direction of our lab to this field. And we went through some hard times in that we really had to change our way of thinking and our training. One of the nice features about this family of genes is that you don't even have to worry about their clinical relevance, because it turns out there's an experiment of nature where several of these genes are actually mutated, and in every case, they are linked to an important human disease. So in case of C2ta that I just told you about, this is linked to immunodeficiency called the Behr lymphocyte syndrome. NOD2 is linked to an inflammatory bowel disease called the Crohn's disease. NLRP1 is linked to vitiligo, which is an inflammatory disease that discolorates your skin, and this protein is linked to a slew of inflammatory disorders, one of which is an inflammatory disorder caused by a drop in temperature. And as a drop in temperature, the person with this defect will start to have inflammatory skin diseases, they'll have fever, and they'll have severe problems with their health. And this has been one of the focus of our lab and many other labs. In addition to that, we have also focused on this one, which when mutated is linked to skin problems that are associated with autoinflammatory diseases such as atopic dermatitis. So all of these actually are linked to human diseases. So like I said, we didn't need to justify our reasoning for studying this family of genes. We did this from a biochemical and molecular approach, and as a result, we look at this gene family, and this again is a composite of many people's work. We found that this gene family has a remarkable ability to target some of the most important inflammatory pathways. So for example, this group of proteins target this pathway that produces something called interleukin-1. Interleukin-1 is an important inflammatory molecule, and it is, for example, related to pain. It's also related to fever. There's another group of proteins that regulate another pathway that causes interferon, and interferon is a key regulator of antiviral responses. There are other proteins that regulate the TNF pathway. I already told you earlier that TNF is the major interfering pathway for the treatment of rheumatoid arthritis. And of course, C2ta is important for the MHC class II promoters. We, in our lab, have established a lot of disease models to look at the effects of NLRs, and we found that these genes have an amazing repertoire of effect on a large number of diseases. This includes bacterial infections, so all of these are done in our lab, where we found they are important, for example, in Staph aureus infection linked to multi-drug resistant MRSA type. It's important for stomach bacteria such as Shigella, Klebsiella, and important for sexually transmitted diseases, for gingivitis, as well as for the famous anthrax, which is a biodefense agent. Viral infections such as influenza and vesicular stomatitis are also regulated, their responses are also regulated by this gene. But in addition to infectious diseases, we found that this gene family is also very important for a large number of inflammatory diseases, such as multiple sclerosis that I already told you about, and sepsis, skin allergies, colitis, fibrosis, pulmonary infections, inflammatory response to for radiation therapy, colon cancer, and more recently, cancer vaccines. So the repertoire of functions that are related to NLRs and by extension inflammation is absolutely huge. And finally, our third exploratory project was identifying proteins that can confer tumor survival. And one of the ways we looked at this was to treat cells with chemotherapy and then look for proteins that are altered by chemotherapy. And through this process, we found a protein called DJ1, and it turns out, and this is where it is, and these are cells that are not treated with chemotherapy, and this is a cell treated with very effective chemotherapy. And we found that this gene actually, when it's highly expressed, it's actually a poor prognostic marker for lung cancer, and other people have shown it's important for ovarian cancer and breast cancer. It turns out that this protein actually interferes with a group of proteins called tumor suppressors that suppresses cancer. In addition, we believe, and we have data to show, that this protein enhances tumor survival when tumors are in a hypoxic, which means there's very little oxygen, an oxidative condition, meaning there's lots of free radicals. So these are conditions that are very prevalent in cancers, and this protein allowed the cancer cells to have a selective advantage. Really interestingly, when we published the paper, there were only 5 papers on DJ1. But in that same year, there was a remarkable paper showing the genetic basis of Parkinson's disease is due to a mutation in DJ1. And we now think the reasons for this is because in cancer there are lots of this, the high expression of this protein, allowing the cells to survive. But in Parkinson's disease, there's the selective depletion and death of a selected group of neurons, and they have a loss of function mutation in this gene, so they cannot survive. So this is one gene that actually intersected two very important diseases. So as you can tell, our joy and our hope, of course, is in the research that we have done and in the people that we have trained. And I don't know that my lab is aware, and I see many of you in the back, that one of the happiest time in the week is Friday from 9:30 to 11:30. I know for you guys, it's time you feel like you're being grilled. And that is our research in progress. So people present their work, and I sit there and I kind of critique what they have done. And the reason that is the happiest time of my week is because that is the time I live through the work of others, those in my lab, and I really get to find out what is the latest discovery that they have made. So for many of us, when we find a new finding, and for that very brief moment, when we think we are the only ones who know of that discovery in the whole world, we hope, that is an incredible feeling. So that is the joy of all of us who are in research. So many of my graduate students, I have to say, have achieved much more than I have when I was at their stage. And many of our postdocs have achieved significantly more than when I was a postdoc. So I have high hopes for the future of science. However, I have to say the very last word of my title is fear. That in recent years, we have noticed that academic researchers have been in a little bit of a funky mood. That is, we're kind of have a lot of angst and anxiety. And one of the rituals, like I said, of me going to different places to give seminars is I get to talk to the students. But at the same time, I also get to talk to the faculty. And one of the things I've noticed more and more is that these faculty gatherings, or when they take me out to dinners, they have a lot of angst and anxiety of what is going on in academic science. I think that several developments in the last few years have really occurred in the form of a perfect storm in that we have to be concerned about. The first is that there is no question that this is the toughest grant environment that I have ever encountered. So for the agency that supports immunology, the grant funding cutoff is at 8th percentile. So to put that into perspective, that means out of 100 grants, you have to beat down 92 grants to get funded. And the 92% that didn't get funded are not slackers. Among that group are people from the best universities, from Ivy League schools, from the top research institutes, from the top public universities, such as us. So there's no question that this is a very tough environment. I'm very glad that NIH actually has the foresight to give young faculty a special privilege, and that their percentile don't have to match those of us who have been in the game for a long time. So I think this is a significant foresight to preserve our next generation. Although at times I wish there was a special allotment for those of us who qualify for AARP. So in addition to that, it is really amazing, I think, that Carolina, we have improved our funding by about 13% in the last year. So I think this is a testimony to our faculty and the collegial nature of all of us. The second problem that we are encountering, of course, is there are less and less support for the institution. Especially for a public university, we are aware that we are in a time of the Great Recession. So again, for my generation, none of us have seen anything like this. When I go out, however, one of the common complaints is that they are worried that the university has really only emphasized how much grant funding we can obtain. And as a result, our research discoveries and our teaching and service are being looked upon as our secondary function. I think those are extremely important functions, and I have to give kudos to our university in that many of these, service, teaching, etc., are still held as very important functions. And I hope that we can continue this. The third, and my most passionate, is the brutal review process of manuscript that all of us who are in the trenches can identify with. And this is mostly in the area of the high-profile journals, and we all know what these are. So this process, we who are senior, or we oldies, are probably used to rejections. So I have counted in my career, I have probably received about 200 rejection letters from all sorts of sources, from grant agencies, from papers, from journals, etc. So for us, it's a little bit easier to take this. But for the young people, this repeated rejection is very hard to handle. So everyone now has horror stories of what it takes to get a high-profile paper published. It is not uncommon for a high-profile paper to go through rounds of submission and resubmission, which take 2 months each. And in between, there's a revision process, which take 2 more months. And at the end, we have the infamous third reviewer who decided to be so nitpicky that he found something wrong in the pages of data that we showed them. And they decided this paper is not worth it for this high-profile place. And so many papers are rejected due to a small point of contention that really doesn't change the conclusion of the paper. So this has become a lottery system that, depending on your luck and depending on, unfortunately, I have to say, your politics, you have a chance of publication in these papers. If this was just a matter of pride, this would not have been a big issue. However, in many cases now, this has become a matter of life and death. So grants, raises, promotions at some places really depends on if one has a high-profile paper. However, it is ironic that many times when we try to publish a major discovery that's completely novel, these high-profile places actually have a very hard time accepting them because they really haven't heard about these new discoveries. So instead, these papers get relegated to somewhere else. So when I look back into my own career, the new discoveries I made are the ones that I had the hardest time getting into a high-profile paper. When I'm with a crowd, then it's much easier to get a paper into one of these journals. So I think in giving high-profile publications such a decisive power in deciding the fate of all of us in science, it's really an abdication of our power and authority. So in a vicious cycle of sorts, papers that are published in high-profile places also are cited more, even if they're not the first paper to be published on that topic. So I really call on all of us to not use a high impact factor as the only means to judge our job applicants. If I were judged by that, I would probably not be here, and many of my colleagues would not be here. I also implore that all of us to support journals that are not considered such high-profile places, to support open access journals, journals that are really managed by real scientists, and in such that we also support our society journals. So I think this is one of my really concerns. Unfortunately, this kind of assault on academic science has obviously influenced our next generation. So I have 2 stories to tell you. In the first one, I was asked to be an advisor for a training grant at a major university. And I met with 10 very talented trainees for this training grant. And at the end, I asked them, which one of you wants to have an academic career? So years past, if I asked that, many people would raise their hands, because in my generation, that was our top choice. If we could have an academic career, that's what we would have wanted. And not a one person raised their hands. So to that, I was truly shocked, and I asked them, why not? And they said, because it is too hard. There are too many things that has to be done that we really don't like, that's unrelated to science. So I think as leaders in science, we really have to be aware of all these assaults on academic science. Another event that really caused me to be shocked was when a postdoctoral fellow of mine, who was really outstanding, was offered a job in an academic research institute, a major academic research institute. He was also offered a job at a major pharmaceutical companies. Again, in my generation, we might not have a hard time choosing, but he decided to go to the pharmaceutical company. And at that point, I understand that we might be in trouble, that we are losing many of our best and brightest to perhaps other places. So I'm really delighted that there are now many choices for our young people to go to. But I'm worried that many of the things that are occurring right now is not good for academic research. And to be honest, I don't have an easy answer to counter this new trend. And I'm delighted that there are many new opportunities for our young scientists. But on a personal level, I strongly believe that if we provide a good environment and show that academic science is really a good thing to be in, and actually a happy place to be in, that we will be able to attract and help budding scientists. One of the retorts I always have to students who decide not to take an academic position is I ask, how many divorce lawyers have you seen get really burned out of their careers? I hope there are not too many divorce lawyers in this crowd, but if there are, I apologize that I use you as an example. And then I say, how many academic professors have you seen voluntarily decide to leave their jobs? I actually don't know of any. So we must have a lot of joy and hope in what we are doing. So I hope to use that as a counter to what they have experienced. So at a personal level, I really believe that good mentorship and support for our young people is of utmost importance. I mentioned during the beginning of my talk that my postdoctoral mentor, Jeff Frolinger, encouraged me to apply for a position here, and that is why I'm here. I did not tell you that he also read my first grant, the dreaded first grant of a junior faculty. And he tore it apart, and then he sat me down and told me why my grant has so many problems. And to this day, I'm very thankful that he took the time to do that. Jeff, in turn, always credited his mentor, Ray Owens, for unparalleled mentorship. Ray Owens was a chair of biology at Caltech, and he was nominated for the Nobel Prize for his work on tolerance. So he was a famous scientist, but his mentorship to all, and even his grant students, people like me, were just really amazing. So my first encounters with Ray was when I was a junior faculty, and I submitted a paper through him to the National Academy of Science. And he handled the paper, and then when the paper was accepted, he wrote back a personal note to me, congratulating me about the good work, and actually talked about the content of the paper. I was truly amazed, and I kept his note to this day. So this simple gesture of kindness from a well-respected scientist can carry such weight and such meaning. It is from this experience that I learned the importance of mentoring and believe that good mentoring can really counter many of the negative assaults on academic research. And it is also my belief that as we become more senior and have more status, that it is our role to really mentor our postdocs, our junior faculty, and our students. So just as I began my talk with a story, I'd like to end it with another story. A couple of years ago, I got a phone call from a person who used to be the dishwasher in our lab. So he was an undergrad who did dishes in the lab, and he was interested in research. So I talked to him and put him on a small project. And from there on, he went to graduate school, got his postdoctoral training, and went on the job market. So he called me up and wanted to ask me for advice. And so for a couple nights, I spent a few hours with him discussing the pros and cons of what he was trying to select. And afterwards, he was appointed a assistant professor at a research university. And subsequently, he asked me to read his grant, and I did that and sent it back to him. And after that, he actually got his grant, and he wrote me a very long note of gratitude. And he said it's at that point that he realized he really can't do anything for me, that he really can't help my career. And his point was that what he learned he had to do was to pay forward. And I think at that time point, when I read his letter, I really sat there for about 5 minutes thinking and reflecting on what he just told me. And I realized that in this former undergrad who was the dishwasher, now I have created a mentor who will hopefully take after people like Ray Owens. So our generation has passed on the mantleship of mentorship. So because of this, I'd like to credit my present lab members, many of whom are here, for much of the work that I described and the previous lab members of the lab. But I also like to close this with a quote for the obituary of Mrs. Norma Berryhill. Of all her accomplishment in that note, it said, the most important is that Mrs. Berryhill served as a mentor, counselor, and confidant to hundreds of medical students' wives. And so her accomplishment is truly amazing, and I believe that it's because of her that Carolina is the way it is, that we are such a collegial place, that it's really exceptional, that many of us have spent our entire career here. So I'd like to thank many of you for coming, to taking the time to come and celebrate this event. I'd like to thank my colleagues, my collaborators, my family, my immediate family and extended family who are mostly on the West Coast and some are actually on the other side of the world, my 2 wonderful daughters and my wonderful friends who have supported me through thick or thin. Thank you very much for your time. Thank you very much.
Thank you, Dr. Ting. As you were talking, I think I speak on behalf of all of us when I say we are very glad that 20 years ago you came to Carolina, and we're so glad that along the way you resisted those many invitations to go elsewhere. Thanks for sharing your exciting science and for demonstrating what everybody has come to know of your winsome leadership style and your infectious enthusiasm. Thank you for inspiring us. You've now joined the distinguished ranks of Berryhill lecturers. I believe you're the 26th such one. All of you, thank you for coming tonight. We have a nice reception. Please come congratulate Dr. Ting, make new friends, and enjoy old friends. Thank you so very much. Good night.